Now we're going to move on to discuss one of the most important and highly tested viruses on step 1, the human immunodeficiency virus, also known as HIV. Do you remember what kind of virus HIV is? It is an RNA retrovirus. Very good. Do you remember the other clinically important RNA retrovirus? HTLV. Here's a wonderful figure of the HIV virus that illustrates the important structural components of the virus. The virus is a diploid virus, meaning it has 2 copies of its RNA genome. There are 3 important genes to know about when discussing the HIV virus. That is the ENV, GAG, and POL genes. ENV gene encodes for envelope proteins, glycoprotein GP120 and glycoprotein GP41. These proteins are found on the envelope of the virus and assist in attachment, fusion, and entry of the virus into the target cell. More specifically, glycoprotein 120 is responsible for attachment, while GP41 is responsible for fusion and entry after the attachment of the virus. A way to remember this is that the number 4 gets you through the door. The GAG gene is responsible for encoding the protein P24. P24 is a capsid protein, meaning it's found inside of the envelope. The way I remember these proteins is to think of the virus as a layered onion, drawn here. It's a 3-layered onion, with the outermost layer being the protein with the greatest number, GP120. This protein is responsible for the initial attachment of the virus to the target cell. Right inside of that GP120 is glycoprotein 41, the second largest number of the proteins, 41, and this protein is responsible for fusion and entry of the virus after it has become attached with GP120. The least and middlemost protein is P24, and this protein is used for diagnosis of the virus. This way can help you remember that GP120 is the outermost and it's used and responsible for attachment of the virus. GP41 is less than 120 and right inside of the GP120 and is important for fusion and entry. And then P24 is kind of the inside capsid protein that's used for diagnosis. Remember, this is just a way to help remember these proteins. It is not actually that GP120 is outside of GP41 because they are both part of the envelope. The POL gene encodes the meat and potatoes of the virus, including the reverse transcriptase and the integrase enzymes. Remember, the HIV virus is a retrovirus, meaning when the RNA enters the cell, it's reversely transcribed into its complementary DNA by the enzyme reverse transcriptase. That DNA is then integrated into the genome of the host cell by this integrase enzyme. And then once the DNA is integrated into the genome, it uses the host cell machinery to then transcribe into mRNA and translate into functional protein for the virus. Transmission of HIV can occur through sexual contact, blood transfusions, IV drug use, or across the placenta and during breastfeeding. HIV primarily infects both macrophages and CD4 positive T cells. Infection and replication of HIV within the macrophages is key for its ability to spread to other tissues. HIV also relies on chemokine receptors, CCR5 and CXCR4. These are co-receptors that help it enter the target cells. The virus will bind to different receptors depending on whether it is entering a T cell or a macrophage. Early in the infection, HIV binds to CCR5 plus CD4 to enter macrophages, where the virus replicates rapidly. Later in the infection, it infects T cells with a CXCR4 co-receptor plus CD4 to infect C cells, and that's when it causes the later stages of the disease. In some populations, there are patients who have inherited a mutation that results in genetic deletions of this CCR5 gene. The patients who are homozygous carriers of this mutation of CCR5 are effectively resistant to HIV infection and have immunity to HIV. Heterozygote carriers will have a slower, milder course of the infection. There are two tests used in the diagnosis of HIV, but before we discuss them, do you remember the difference between a highly sensitive and highly specific test? So we're not going to get wrapped up in the details of biostatistics here, but it's important to remember that sensitive tests are ones that are used to rule out disease. If a sensitive test is negative, then this patient likely doesn't have the disease. Okay, the sensitive test is used to rule out the disease, and we can remember the mnemonic SNOUT. Now for a highly specific test, it is used to rule in disease. It is used to rule in disease, meaning that if a highly specific test is positive, then the patient most likely has the disease. So we could use this mnemonic SPIN to remember that specific tests are used to rule in disease. So with these rules in mind, we can remember that diagnosis typically starts with a highly sensitive test, and if that's positive, it is confirmed with the highly specific test. Very good. Diagnosis of HIV begins with a highly sensitive test or screening test called an ELISA. Do you remember what ELISA stands for? So if we think back to our biochemistry, it stands for Enzyme-Linked Immunosorbent Assay. This test detects the presence of HIV antigens P24, P17, GP120, or GP41 in the patient's serum. This is a very sensitive test, and as we said earlier, if this test is positive, then that does not necessarily mean the patient has HIV. It just means that we need to go ahead and do the confirmatory test, which in HIV diagnosis is the Western blot test. The Western blot test is another test used to detect protein in the patient's serum, but it is more specific than the ELISA. If the patient has both the ELISA and the Western blot positive, the patient most likely has HIV. So the two diagnostic tests used for HIV infection are the ELISA and Western blot assays. There are two periods of time that these protein tests, the ELISA and Western blot, are not useful. The first time is within the first 2 months of disease. The reason is that within the first 2 months of an HIV infection, the viral proteins aren't produced at significant enough levels for the ELISA and the Western blot to be effective and detected. The second time is when babies are born and tested when they're born to infected mothers. So if the mother is infected with HIV, using ELISA or Western blot to detect HIV transmission to the baby isn't necessarily the most effective test because GP120 can freely cross the placenta and can be picked up in the baby's serum even though the baby may not be infected with HIV. It could just be the protein from the infected mother. Once the diagnosis of HIV is made, the polymerase chain reaction, or PCR, is performed to determine the patient's viral load. The viral load is the amount of viral RNA that's present in the patient's blood serum. Not only is the patient's viral load used to determine prognosis, it is also used to follow up efficacy of drug treatment. The CD4 count is also used to assess and monitor treatment process. So we can think of the PCR and CD4 count as responsible for monitoring therapy, while the Western blot and ELISA are the diagnostic tests. Remember, HIV infects CD4 cells, and the number of CD4 cells present in the blood is a marker for how well controlled the infection is. So the next important thing to cover is answering the question, when do we diagnose a patient with AIDS, or acquired immune deficiency syndrome? We all know that HIV leads to AIDS, but when is this diagnosis made? The diagnosis of AIDS is given when there's a documented HIV infection along with one of the following criteria: a CD4 cell count below 200, an AIDS-defining illness, and or when the CD4 percentage is below 14%. We're going to discuss what AIDS defining conditions are upcoming in the next slides. Now time for a flash quiz. What hepatitis B antigen is significant for high transmissibility of the virus? That would be hepatitis B E antigen. Remember, the E antigen stands for excited hepatitis virus. It is excited and is highly replicating and therefore is highly infectious. This graph shows the course of an HIV infection in individuals who are not treated with antiviral medications. We can see that this graph has a double y-axis, with axis on the left side illustrating the CD4 count as represented by the red line, and then on the right we see the HIV RNA copies per milliliter, or the viral load, represented by the blue line. The x-axis represents time in months and years. We can see, as a general pattern, the CD4 count drops steadily and the HIV viral copies increase over time. However, if we look more closely at the graph, we notice it isn't a linear or exponential change in the viral load and CD4 count over time. There are some peaks and valleys that illustrate some important concepts. The first thing we notice is that there is an acute and a chronic HIV infection. The acute infection involves a viral load increase exponentially, as seen here, with dissemination of the virus and the seeding in lymphoid organs. We also notice an initial drop in the CD4 count by close to 50 cells per millimeters cubed. During this period, patients will experience a flu-like syndrome, fever, lymphadenopathy, malaise, headache, and myalgia. This is known as acute HIV syndrome. Now, after the acute HIV syndrome, the body's immune system actually begins to fight and win against the HIV virus infection. We can see here that the viral load decreases pretty significantly and the CD4 count bumps up a little bit more in response to our immune system properly or adequately reacting against the virus. Now, during this time, the virus then enters a period of clinical latency that can last for years. Even untreated, it can last for years. But we can see that over 5 years, even with the viral load staying pretty constant, the CD4 count steadily is decreasing. It isn't until the CD4 count drops to about 200 when we begin to see the viral load start to pick up again. Remember, the CD4 level diagnostic criteria for AIDS is less than 200. So we see here at 200, that is the time when AIDS is diagnosed and the patient will begin to experience the dreaded opportunistic infections that will ultimately kill the patient. In addition to the opportunistic infections as the CD4 count declines, the patient may experience a variety of constitutional symptoms such as weight loss, fever, and chronic diarrhea. A good way to remember the progression of HIV is to think about it in 4 stages that all start with the letter F. Number 1, flu-like. Number 2, feeling fine. Number 3, falling count, CD4 that is. And number 4, final crisis.